Hello. So this is the drug development chapter for Strider. So we're going to be talking about having a really broad overview for the drug development process. Not only the discovery of candidates, but also the phases that they have to go through to get FDA approved. And so this is a nice little chart. It's talking about what the drugs target. Whether they be enzymes, the G protein coupled receptors, which are one of the major classes. This was actually these were discovered by Lefkowitz at Duke University. He actually won the Nobel Prize a couple years ago for this discovery, as well as some transporters with that. So this just sort of gives you the broad range of different targets that drug companies go after. So the process is usually either one of two ways, sort of a very broad scheme. You have a compound and you notice that it has a physiological effect. From there, you try to discover the molecular target so you can understand the drug. So this would be the sort of classic sort of natural products. So folk medicine would say, hey, there's something, if you drink this kind of tea for this kind of condition, then you can go in and isolate the compound and see what it goes after. Most pharmaceutical, the other way you can do it is to say, okay, we know that if we knock out this, if we inhibit this enzyme or receptor in the on a tumor cell, so we can stop tumor growth, and so you know what the target is. Now you got to find a compound that inhibits this, and then you get it, and then after you find the compound, again, you measure, things like tumor growth or whatever, so that the physiological effect. These are generally how, sort of at the very highest level, this is generally the two ways that you do this. So for drug development, it's a lot of this is going to be about how well the drug works. Okay, so the first part of this is looking at what are called KDs, so the association constant, and so this is very similar to the KM, where it's usually you look at 50% of the receptor bound, and so KD is receptor times ligand divided by receptor bound, and so these are typically given in a concentration. So what ligand concentration is half the receptor bound. So if you have a biological response, so how how many tumors die, or some sort of tumor cells die, stuff like that. So you'll get this. Hopefully, you get this dose response. So as you increase the dosage, so at this low concentration here, only say, so only 10%. If you're at this dosage here, only 10% of the cells die, but at this one 50% die, at this one it's 90%, and so on and so forth. And usually you get either EC50 or EC90, something like that. And so at what concentration does half the cells die, what concentration does 90% die? And then you rarely ever do 100 because you can't always get to 100. So it also, what is 100? Is it 90? Is it 99? Is it 98? Is 98 okay? You have to be 99.9, 99.999, whatever. But we can estimate what 90 would be and what 50 would be quite easily. So you see this one is just if you see EC, it's effective concentration. The other thing would be inhibitory concentration. So if you have a receptor, say, you'll get these curves. So enzyme activity, with that, and so at low concentrations, you sort of measure the activity, with that, and so you'll get these curves here. And so hopefully at increasing concentrations, you'd be able to see enzyme activity, so see improvement. So one of the things you have to do with with drugs is be able to target something specifically. You want to target your receptor. You don't want to hit something else, right? You don't want to cure cancer, but also destroy their liver. It's not going to do you any good. Things like that, or give them a heart attack, a lethal heart attack, and stuff like that. And so you have to be careful. And so this is a panel, stuff like that. And so The dark blue shows no activity, but these are a bunch of different receptors. These are a bunch of different compounds, ligands. So could potentially inhibit receptors. And so the darker the red, the more likely it is to target here. So if you're trying to inhibit this one, right, you'll also hit this one, this receptor, and this receptor. So this compound hits a bunch of them. Okay, and so you can have these off-target effects. So this compound would not be very selective. But if you're looking at, say, this one, right, and you go across here, this compound D only inhibits that receptor. So that's going to be very specific. Same goes with this one. If you go across, right, so E, if you go across, I'm pretty sure that's down one, right? This is another one that's very selective, like that. And so you don't want to hit, multiple receptors. If you do, okay, if you do have, at say, a higher concentration, that's fine, as long as this gap is quite high. So maybe higher concentrations you hit B, C, and D, but at lower concentrations you are selective for A. And the wider the gap, the better that is, because of differences between people and metabolism and things like that. So the less likely you are to get into these ranges where you're going to start potentially having deleterious side effects. And so you do have to be careful with this. So there was an issue about 15 years ago with Vioxx, in that it was a great pain reliever, so with that. So that class of drugs, my boss at the time was taking it, and he said it relieved pain that I didn't even know I had. So with that, but the problem is Merck went in and they started noticing a few years earlier that people with this that were on it for long term were having heart attacks. So with that, and it wasn't. What is it? They say it's not the crime, it's the cover-up. Had they told the FDA right away, everything probably would have been fine. But they kept it a secret because they didn't think it was that big of a deal, and they wanted to keep the sales up. And so the FDA got involved, and everything, bad things happened. Stuff like that, and so so the FDA got much more stringent about looking at these off-target effects after that. So one of the things you have to do is, once you have a potential compound, you have to look at what's called the ADME, and that's the absorption, distribution, metabolism, and excretion. So after you take the drug, after you take the pill, what happens to it? And so that's what this metabolism and the ADME comes into play. So if you're going to take a pill after you swallow it, so this is the absorption, and so there's this Lipinski's rule of 5. Sometimes it's called the Pfizer's rule of 5 because he works at Pfizer. He was a medicinal chemist for them, and so this isn't a rule or anything else like that. It is simply something that he had noticed and he had published on, and it's not a Bible. It's not a law. It's just guidelines. That's things that he noticed. And what he noticed is that if you want to have a good drug that has bioavailability, okay, you don't want it to have greater than 500 molecular weight. So with that, more than 5 hydrogen bond donors, so these are like NHs and OHs and things like that. Acceptors more than 10. So with that, and you don't want a partition coefficient, a log P, greater than 5. Okay, did I talk about log P? So log P. So this is log P is the log of your drug in octane, your solubility of drug in octane, divided by the solubility of the drug in water. Okay, so a log P of 5. What that means is that it is really, it's 10 to the 5th over 10 to the 1, or 10, or 100,000 to 1. Okay, and so what this is saying is that if you have, for every one molecule you have it in the water, right, in your bloodstream, 100,000 are going to be precipitated out, okay? Because octane is obviously very, it is going to be, very hydrophobic, right? So if it's this here, it's just not going to be soluble enough in the bloodstream in order to be able to work well, okay? And so you can't have it too hydrophobic, or it's just not going to work real well. Okay? And so one of the classic ones that works really well that follows this is morphine. Okay, so the molecular weight is less than 500, right? So it's 285, right? So in this case it's 285. Okay, so you can see this. The log P down here is 1.27, right? So it's about 20 to 1. Something like that, and so not too bad. Number of hydrogen bond donors, right? So you've got an OH here, you got an OH here. So you got the 2. Hydrogen bonding acceptors, right? So anything that can take the lone pairs, okay? And so these are actually also acceptors as well, because they have the lone pair, right? And then you've got also have one here. So there's 4. So for here, there's 4 of these, and then there's 2 of these. And so, well, this follows Lipinski's rule of 5 quite well with that. And so, right. Okay, so distribution. Okay, so where does it go? And so one of the things, so not only, once it gets into the bloodstream, where is it going to travel? Where is it going to start to accumulate? And so one of the things that you can do is you can make radio-labeled versions of the drug. So you can make, say, a carbon-14 or, oxygen, what's that, 18 or some sort of radioactive version of this drug, or something that would light up in the isotope that would light up in your machine. Okay, and so you can sort of scan your brain, heart, liver, kidney, muscle, some of that. And so if you notice, right, so for that, a lot of it's going to get, in this particular drug, a lot of it's going to get accumulated into the liver, a little bit into the kidneys, right? So this is how you break it down. But you are seeing some inside the heart, which is fine if it's a heart medicine, some of that. But you are also seeing a little bit in the muscle, but it doesn't get into the brain. So you can do this, right? So if it's radioactive, it's not going to, it's going to have the similar properties to it, but you'll be able to detect it. And so what your body really wants to do is it's really, really good at getting rid of things. Okay, so you see these sort of like juice cleanses and things detoxing and stuff like that. You might as well just be throwing your money in a hole, stuff like that. So this is what your liver and kidneys have been evolved to do. If you need these things, if you actually need these things, you need to talk to your doctor, because you have potentially kidney failure or liver failure or something is wrong, stuff like that. So go and give that money to a charity, because it's going to do a lot more good. All you're doing is making somebody rich. Okay, so what your body really wants to do is it wants to make the compounds more soluble. If they can become more soluble, you can get it into, you can then excrete it and stuff like that. And so so there's several different ways it can do this. You can use these cytochrome P450s, stuff like that. So it's 450 because it's they're iron bound, and so inside of a porphyrin, and so they absorb at 450 nanometers. That's what the 450 stands for. But what they do is they oxidize with that. And so here what we're doing is we're taking this taking this H here and turning this into an OH. Now OHs are a lot more soluble, alcohols are a lot more soluble than alkanes. So now we've made this compound just a little bit more soluble. If it's more soluble, then we can get rid of it. Okay. But that's the other things it can do is it can add groups onto it. So this is a glutathione, right? So you can add sugars. So in this, I believe this is glucuronic acid. Yeah. So excuse me, this is gluconate. So this is like glucose, but it's an oxidized version of glucose. Now this molecule is a lot more soluble, even though it's pretty soluble before. The other thing it can do is it can add sulfates. You can add phosphates, you can add, other sorts of things. But basically what you're doing is you're making these a lot more soluble and a lot less active too. Some of that. So you can block the activity, things like that. And because your body, think about this, 5,000 years ago, right? No medicine, your body had to do everything. And so people, so your body has evolved to do this really well. Okay, so with metabolism, right? So one of the things we have to do is we do have to measure how quickly this breaks down. Okay, and so after you take the drug, how quickly there's going to be a peak time for when it goes and dissolves into the dissolves in the gut and disperses out into the bloodstream, stuff like that, and then your body will start to metabolize this. Now you can do a whole bunch of things to change this, right? So if you get different formulations, you can get this either as a longer curve. So if you have an extended release, you would have this would be flatter. You could have you could do make the pills. There's like laser coated, laser drill pills, or that you can come in and you can the coating, and so it releases really fast, stuff like that, and so you can play with this. So scientists can come in and play with the steepness of this curve, but you need to know this, because if this is really high and then really low, if your body metabolizes this really fast, you may have to take the drug multiple times a day to keep the concentrations up. So it's not preferred. 3 times a day is really compliance is tough. Most drug companies try to go for once a day because people are pretty good about that. Twice a day is kind of iffy, but they'll do it, stuff like that. But once a day is preferred. So how do you get rid of it? Well, there's 2 different ways. There's the urine and the feces, right? So with that, and so when you're doing animal model, right? So with that, when drug development with animal studies, right, you have to collect it, but also with human studies, with that they'll collect the urine and the feces and track where it goes. Again, this is another one where if you have a radioactive compound, you can track where it's really easy to track. It's like a Geiger counter in there, and you can see what how much is in the feces, how much is in the urine, and things like that. So you can see where it goes. So and you also have the half life, right? So how long does it take before half the drug is gone? Okay, they need to be able to understand that, how quickly you get out of this. So for that, as part of this, one of the things you have to worry about, and they actually will come in, and there's a huge effort after this, is to take a look at the actual metabolites, what your body does with the drug. Do those metabolites, are those toxic? And that can be a huge issue for some of the drugs. And so one of the ones that more common ones we have to worry about is acetaminophen. So this is Tylenol. This can actually get in a small amount of in a portion of the population. This will get oxidized to this form, and you'll get glutathione on here. And it turns out that this is this is a toxic to your liver. And so you can literally overdose on Tylenol because it destroys your liver. Stuff like that. So there was an issue a few years ago where they had infant Tylenol and child's Tylenol, and people would grab, you got a sick, a feverish kid at 2 a.m. You're a little tired. If you grab the child concentration of Tylenol, you could overdose your kid, your infant, because you would, I need a teaspoon. Well, but if it's a lot higher concentration, you would overdose. The child would overdose and destroy the liver. So that's why you don't have infant Tylenol on the market anymore. But it is a big issue. So there is what's called the therapeutic index, and it's the ratio of what the lethal dose versus the effective dose. And hopefully the wider this is, the safer the drug. Okay, so for that, because the less likely you are as somebody takes the drug, it's going to build up to toxic effects. Now it depends on the drug, right? So if it's a an anti-cholesterol drug, and you're going to be on this for the rest of your life. You want this as wide as possible. The FDA is going to be like, you have to make this as safe as you can. But if it's an anti-cancer drug or that, this can be shrunk a little bit because the FDA will tolerate a smaller shift, a therapeutic index, because right, you're going dying of cancer, right? There's immediate concerns with that. So plus it's given under a much more controlled environment usually. So if you look at the therapeutic index for a bunch of different psychoactive drugs and some of that, so you can see and notice how this is a what is it? This is a logarithmic scale. So marijuana, so for example, is you're very much not going to the likelihood of you dying of smoking too many joints is pretty low. So that especially it's a 10 times safer than caffeine, stuff like that. So and even ecstasy and cocaine, things like that. So there's 10. Stuff like that. So, but if you notice the dependence potential here, so these are low, these are high. What's really tough is things like morphine and heroin, stuff like that. So not only are they do they have a low therapeutic index, okay, especially heroin. I think it's like 3 to 1. It's like 3 to 1. But it also has an incredibly high dependence potential, and so that's why we're having such issues with the opioid deaths, is because people build up a tolerance, so they get higher and higher, so they take higher and higher concentrations with that, and so they can very easily go over into a lethal dose because the gap here is so low, so with that, and so so there is a wide spectrum for these things. So how do we do? So how do we discover the drugs? Well, one of the best ways to do this. Well, I shouldn't say best ways. It's not a great way to do luck, stuff like that. But one of the more amazing ones is just pure serendipity. So the greatest words in science is not eureka. It's oh wow, that looks weird, or that looks funny, and then I wonder why. Some of that, and then it's not just that you see something that's funny, but then you follow up to try to find out why. Okay, so penicillin is the classic example. So Alexander Fleming was he was a really sloppy microbiologist, some of that, and he had left some plates out with some mold on there, and he came back. Came back on Monday, and noticed that there was some around the mold, and then there was clearings around. There was bacteria growing on the plate, but there's also mold, and then there was a ring around that. I'm sure other scientists had seen that phenomenon, but they had just said, "Oh, heck with it," and just discarded it, or didn't notice it, things like that. But he went in, and he said, "I wonder why," and tried to figure it out. And obviously, he did, and then he discovered penicillin, which revolutionized medicine. So with that, and so to being able to understand why something happens, that's the important part. Something looks weird. And why is that? Okay, so if you have these two psychoactive drugs, right? So with that off-target, well, dopamine. I'm not entirely sure what this one, chlorprozamine. I forgot what that one is. So with that, and so these are off-target effects. People have noticed, when they give drugs, something else happens. So with that, so somebody's observant. So with that, so the other thing is curious side effects. So the classic one is sildenafil, which is Viagra. This was initially developed by Pfizer as a as a heart for heart disease, and actually it is approved. It's not a great heart disease drug, but it works. But they somebody over in the UK at a clinical site, they had noticed a curious side effect and noted it, and when people were starting to sort of people were given a heads up, they noticed that this was happening more and more, increasing side effect. And so they realized, oh my goodness, this is, what do we have on? We may have a drug for, for erectile dysfunction. The other one is, another one like that is Latisse. It's a prostaglandin. You can notice that because of the prost here. But also, it's one of the few times you see a pentene in the in molecules, and there's cyclopentenes with these two long chains coming off of it. That's a classic prostaglandin, and so this one here. So this was used for originally looking for this for glaucoma, um, and so you would give the eye drops. Well, one of the things that they notice, right? So when you give eye drops, they, so you put it in there and it kind of splashes around, right? A little bit's going to get on your, eyelashes and stuff like that. And so one of the things that happened was some of the people were noticing that, just made an observation of, oh my goodness, my eyelashes have gotten so, so nice. So that they've gotten really full and bushy, and it turns out that it actually grows eyelashes. There's actually a condition where you don't have eyelashes, something like that, or you don't have, or they're not very full, and we actually got FDA approval for that. Now you can use it for other things, cosmetic things, now that it's approved. But again, that was one of the, another one where it was like, well, that's weird, and then they followed up to try to figure out what's going on. Okay, another sort of classic one is natural products, and this goes into vogue and out of vogue. It goes in and out of fashion. There's some really important ones, right? So salicylic acid, and so people that's folk medicine, right? So if you've got a fever, you give somebody tea, birch bark tea, something like that, and it turns out that it's the salicylic acid. And so this was originally extracted and isolated and identified. But what they noticed, and so they started selling salicylic acid. The problem is it prevents your stomach lining from regenerating, and so you get these large ulcers to build up. But they notice when you acetylate it here, some of that, and so it still retains that the activity, but you don't have the issues. And so they noticed that, and so once they start looking at the mechanism, they noticed that it hits the cyclooxygenases 1 and 2. So COX-1, COX-2. COX-2's involved in the inflammatory response, which causes the pain, and so now you've got Celebrex and Vioxx, things like that. And so they come into a much more drug-like compound, which hits, in this case, it's COX-2 selective inhibitor. It turns out, though, that if you don't hit COX-1, you have issues with heart attacks. The other one is the statin class of drugs, and so the first one that was isolated is Compactin. So that they made it slightly better, more drug-like by adding a methyl group here, and this was the original sort of statin with that. And so these help lower your bad cholesterol, and then you would come in here and so with that. So and then the next generation was again more drug-like, but it retained these properties here, but they all have this part right here in order to be able to work. It's just this hydrophobic part down here. The other way that you can do this is through high throughput screen. I'm saying like, okay, so early in say the 70s and 80s, they said, okay, we've got all these crystal structures. We should be able to completely design the drug to fit into this. And then they were having issues with this, and so the pendulum swung the other way and said, okay, we know what the target is. Let's just throw hundreds or millions of compounds at tens of thousands of compounds at this thing, and all the different ones, and we'll see. We do a little test to see what shakes out, and then we can get sort of a foothold for what things will go into this. So if you've got you've got a compound library of say 10,000 or 50,000 or 100,000 compounds. They're all different types. So with that, you've got your cells. You've got for that. So you disperse this into a microplate, usually either a 96 well or 384 well plate, and then you run. So you run your assays. Does it kill tumor cells, or something like that, or does it activate a receptor? Does it who knows what it does, or kill a receptor? So you then come in and do some sort of readout, and then you look at you get the data. Use data processing, and you'll notice, like for this one, this compound, these two compounds may light up, but the rest of them don't, and these ones over here are just controls. So it's just throwing things out there and seeing what sticks. with that. And these things are amazing with that. So you can have this sort of central arm that can come in and take it from place to place to be able to do the assays. I've also seen it where they're like a little conveyor belt where the robot arm will take it and then go do something with it, put it back on the conveyor belt, slides to the next. There's a little assembly line. These things are really cool. Some of that to see in action. Okay. The other thing you can do, this one along with the high throughput screening, is what's called combinatorial chemistry. And so this one is where you're looking at, we think this part of a molecule is an interesting sort of, you may need to put something here. And so what they'll do is, so let's say you've got a methyl group here, right? And so let's put, so this is part of your compound, right? Well, what happens if we instead, let's say we put an ethyl group here. How does that change? Or if we put, a butyl group. Or an isopropyl group, or something like that. So in this part of your, CH2, oops, CH3. How does that change the molecule? Okay, and so you can make these slight changes and see, does it make a difference? The other thing you do is what's called a split pool. Stuff like that. And so you would take your original compound and then you split it into three different groups. One of those groups would get a yellow, a yellow portion attached. Another person would get a blue portion attached. Another pool would get the red attached. Then these things would then get jumbled back up. And then now you've got, and then now in this pool, right? So we've got representatives from each one. These get split. Now these guys get a yellow. These guys get a blue. These guys get a red. And so you can come to that when you pool these things together, you've got 9 different combinations of these 3. And again, you run these through high throughput screening and see what, if any of these 9 light up in your assay. Okay. And these things can make it can make a big difference for that, just having a simple methyl group here. And so these are some from a paper from a few years ago of showing just one little methyl group, how big of a difference it can make. And so putting a methyl group here, right, boosted it 200 fold, this one almost 500, this one 600, and this one, just putting a methyl group here, going from no methyl to methyl, boosted the efficiency, more than 2,000 fold. And you say, well, that's an easy change. But the thing is, think about all the places you could put this stuff. What sort of combination is it that puts it in there? And the numbers are daunting. And so are you lucky enough to be able to figure that out? And that's the billion-dollar question, right? So after the combinatorial high throughput screen rage, things kind of came back and said, okay, the pendulum swung back to saying, look, we have all this information, but we also have these compounds, so that we know how to do high throughput screening, but we know how to, we have the structural information. Let's combine this. So instead of doing high throughput screening, we do what's called high content screening. So you screen fewer compounds, but you get better information about it. And so that, and this is also the rise of computational power, and so you can do a lot of this stuff actually virtually. You can do virtual screening with that, but you can go in and make much better choices initially, so you can get better stuff out. So for here, we're adding stuff to this part of the compound, and if you look down here, so these are the compounds that are the side chains that we're adding here, not just methyl ethyl, whatever, methyl ethyl butyl futyl. Things like that, we're adding much better ones. And so you can look at the IC50s, and so this is the inhibitory concentrations, and they're all pretty good. Usually you want to get nanomolar or below, low nanomolar, maybe 10 nanomolar or less. Subnanomolar is even better because that means you have to give less of a dose. And it's the lower the dose you can give, right, the less likely you are to have these off-target effects. You look at log P, something like that. So this one here is getting pretty close to that 5, something like that. And then the bioavailability. So what's the maximum concentration in the plasma? And you'll notice how, if you're um, at these really high log Ps, you don't have a whole lot of bioavailability in the bloodstream. This isn't so bad, stuff like that. But if you're adding this group, it's not. It's actually the least of these four. It's the lowest potency, although it is still really good. It does have a really high bioavailability, and so you have to balance this. It's not just which one's the most potent, because if it's potent and it's not bioavailable, it doesn't matter. Stuff like that. So you had much higher bioavailability, stuff like that. So good potency, stuff like that. So if you put this on there, it's Crixivan, which is an anti-HIV drug. So again, if you notice, if you've ever noticed the ends of these of the generic name, so Vir means it's targeting it's targeting a virus. If it's a nib, it's targeting a kinase. If it's a prost, it means it's a prostaglandin. If it's a mab, it means it's a monoclonal antibody. A statin means it's going after the cholesterol. And so these generic names can actually give you some information. So Vir means it's going after a virus. You can look at genome analysis. So you're not only looking at targets, so identified targets, sort of, the reason. So if you get one class of people that have, so one of the things with the statin drugs, it was looking at a particular sort of family of people that had really high cholesterol, and they were trying to figure out why it is that they had that, which ended up helping to lead to these statin drugs. Stuff like that. And so you can look at, what are the differences between sick people and healthy people? Can you figure that out? And it's not always easy. It's rarely easy. Stuff like that. But then also, with that, and so you can come in and figure this out. And so, for hypertension and these sorts of things, you can figure out the new targets. So lots of times what happens is if you find a target, it'll initially go into animal models, stuff like that. And so typically it's going to be gene knockout. Sort of you're going to be playing around with the genes within mice, that. And so sometimes you just have to give just the regular standard mouse, stuff like that. But lots of times you have to you either upregulate a gene or downregulate a gene. So you have more or less of it, so with that. And so you also have to do toxicological studies with this, and it's usually mice and rats and dogs. So when I worked at Pfizer, you could when down the stairwell, you could hear the dogs playing, so with that. So they sound like they're having lots of fun. But the FDA does require it, so with that. And so if people would say, hey, let's just get rid of all animal testing, well, it's not. It's not practical, and also the FDA requires it. So you would have to be a change in regulation for drug approval, because you have to have that data before it can go into humans. The other thing you can do is you compare the genomes of a pathogen versus the host. So, but that you can also do this in plants, right? So that's what the if you think back to what is it, the herbicide, right? So with that, so it takes out the it takes out an amino acid, so with that, so the Roundup, so with that, it takes out it inhibits a pathway that's not in us, but it's in corn, or it's in weeds, I should say. So with that, but you can also look at antivirals and antibiotics. So this is a SARS protease inhibitor. So this was actually made a few years ago. It's very relevant now with all the COVID-19 issues. So with that, and so this one has a specific inhibitor to it. This is the first SARS-CoV-1. With that, and so this protease is not found in humans. So with that, so that's a target. You can also do HIV, a lot of protease inhibitors, and other sorts of drugs like that. They target those things. Antibiotics is another one. Penicillin is a classic case. Penicillin targets a pathway that's not found in humans. That's why it doesn't affect us. Okay. You can also look at compare genetic differences. If you look at the phenotype and genotype between different classes of people. you can see how it would change the different effects. So this is health disparities. So with that, so why do some some people, so typically why some races are more affected than others is because some people, the population is more likely to have these particular genotypes and phenotypes compared to other places, something like that. And so you can have some pretty big effects, some of that. And so this actually gets into why it's so important to have a diverse pool of people when you do clinical trials, right? So with that, so you can understand these differences, so you can note these differences during the clinical trial, so that. So with that, and so the other thing you can look at is toxic side effects, right? So with that, so small amounts, so 6 milligrams purine, with that. So I believe this is an anti-cancer drug, I believe. So with that, so it can get methylated here, and so this is a toxic metabolite. And so it's either that or it's an antiviral, one of the two. So with that, and so you have to be careful because be able to identify the people that could cause these toxic effects. Okay. Now, once you've discovered the drug and you have gone through animal testing and everything looks good, the FDA scrutinizes your data. They say, okay, we're going to start these gates, and so these different phases. So with that, so the first phase, so to get from preclinicals, so from the point at which you just figure out the target to when you actually get approval, it's typically 10 to 12, 15 years, depending on how long things go and how lucky you are. Stuff like that. So all of this stuff, the preclinical stuff, this is in animals. The phase 1, 2, 3 is always going to be in humans. Right now, it's looking like it's it's about $1 billion, but that may be pushed a little higher, and it's kind of all funny money anyway, stuff like that. So I mean, it's not. I mean, it's real money, but how you account for it is always tough. But most of that money is actually done in phase 3. So phase 1, this is in healthy volunteers, and so these are if you see the ads in I guess it used to be newspapers, but now it's online for volunteers for drug trials. Some of that you see it around on our in Durham. Because there's several companies that do this, but they'll look at for healthy volunteers for that, and they get these sub-potent doses. And what they're looking for is how it metabolizes actually in humans. Are there any weird effects, things like that? It's all about safety, things like that. So it's going to be a small number of healthy volunteers. After that happens, then it goes into a small number of sick people, and it's determining not only the proper dose, is it the 10, the 12, the 10, the 20, or the 50 milligram, which one actually does work the best in sick people, but also does it actually seem to make people better. So this is you get what's called a proof of concept milestone to say, yeah, it looks like it's working. Then also, yeah, we think maybe 50 we get better outcomes with 20 milligrams than 10, but 50 works about the same as 20, so let's go with the 20. Things like that. Once you get to phase 3, this is where you get the big placebo-controlled. Okay, so this is a large number of sick people. Something like that. And so for here, they're what are called double-blind. So you're either going to get whatever the normal treatment is plus the drug, or normal treatment plus a placebo. So a placebo would look exactly like the regular drug, and so the doctors nor the patients know which one you're getting, right? So that you get I participated in one time, and you just got a little card, and it had little blister packs, and you would take the drug, and they would scan it, and we had no clue if it was drug or placebo. And sometimes it actually flips. Sometimes they'll give the drug to people for a while, and then they'll switch to placebo and see if there's differences in effects. But you really do not know. Nobody knows who it is. Somebody's got there's a little committee that goes and takes a look, and they've got the master list of who's getting what, but otherwise you don't know. And the trials can take anywhere from 1 to 5 years, depending on how, what effect. If it's an antibiotic, you're going to know pretty quickly if it works or not. But if you're looking at trying to prevent heart attacks, right, you're going to have to wait a long time because to get the statistical power to be able to do it. One thing somebody told me was that if you have to use statistics to tell if it's working, it's not working, stuff like that. And so for that. So now this is when it gets approved after phase 3. But again, you're only looking at a limited subset of the population, not looking at the whole, world population. And so lots of times, they'll ask, the FDA will ask you to do a phase 4 trial. And so what you're looking for there is just monitoring for crazy side effects in the much larger population that you couldn't get from the phase 3. Some of that. So it's about safety. Now, one of the things that can happen is drugs can sometimes stop working. The classic example is antivirals and antibiotics, right? The molecule evolves, it gets a mutation, and and then those either microbes or viruses can now survive because they're not getting tamped down. Some of that. So the same thing happens with cancer, right? So a new mutation arises in a cell that's not resistant to that is resistant to the treatment that you're giving, and so that one can start growing. It's all about sort of think about it as kind of Darwinian evolution, same with that. So now the other thing that can happen is actually psychiatrics or psychiatric drugs. Sometimes they just stop working, and nobody really knows why for these things. So sometimes it's we don't know. Other times it's simply a mutation and what is it? Mutation and evolution standpoint. Okay. Thanks, everybody.